What was a very interesting finding of these organ-on-chip models is that we could observe that Candida albicans forms microcolonies on the intestinal mucosa, and these microcolonies right below them, they disrupt the tight junctions of the intestinal epithelium. Hello, my name is Gresnigt. I'm a junior research group leader at the Leibniz Institute for Natural Product Research and Infection Biology in Jena. And I focus on host pathogen interactions during infections with the yeast Candida albicans. So we try to study these immunology of fungal infections to try and understand how infections sometimes go worse in some patients, and in other patients infections are efficiently cleared. Using organ-on-chip, we try to elucidate how different immune environments, or for example different polarization of immune cells, is changing the outcome of infection. So in this way we can start to study how specific modulations of host immunity, which is disturbed in these patients with these severe infections, can also be used as an immunomodulatory therapy. In 2019, we started to collaborate with Alexander Mosik from the University Clinic in Jena, where we adapted his intestine-on-chip model to start studying how Candida albicans is inhibited by lactobacilli that are commensal members of our gut and protect ourselves against infections. And from there onwards, we now start to implement these models to answer questions in terms of host defense and interactions during infection. We have adapted this intestine-on-chip model where we can look at the initial interface of an invasive Candida albicans infection, where the fungus is invading through the intestinal epithelial barrier to gain access to the bloodstream. And what we now have been doing is, because Candida albicans can also cause infections in other mucosal niches, such as the vaginal mucosa, we have developed now a vaginal on-chip model where we try to model how the fungus is attacking the vaginal mucosa and how this is eliciting inflammatory responses that may not be beneficial towards the clearance of the infection, but actually contribute to the symptoms of the disease. What was a very interesting finding of these organ-on-chip models is that we could observe that Candida albicans forms microcolonies on the intestinal mucosa, and these microcolonies right below them, they disrupt the tight junctions of the intestinal epithelium, gaining the fungus access to invade deeper into the tissue and eventually reach the bloodstream and cause infection. And this is manipulated by first-line antifungal drugs such as, for example, caspofungin. What we see is that organ-on-chip models, they fill a crucial gap between traditional in vitro models and animal models, where we can start to answer research questions that are not straightforward to address in animal models, simply because the manipulations and the controllability that we want to have is not unifiable with working in a live animal. But on the other hand, when comparing to traditional in vitro models, organ-on-chip models offer increased tissue complexity and a more near physiological tissue architecture to better model the infection processes. So we have noticed that upscaling organ-on-chip models is hard, because you have to simultaneously monitor several of these chips that you need to make sure that the media is constantly perfused. And one of the challenges with these infection models in the intestine is that we use a linear perfusion to remove any of the fungi that did not attach and invade the intestinal epithelial barrier. By using this perfusion system, of course, our cells stay happier, it's more physiologically relevant. However, this can also lead to draining of reservoirs, which is a challenge when trying to perform these experiments. So one of the dreams that we have is to be able to model invasive candidiasis, a life-threatening fungal infection, fully from the start of the infection at the interface of the intestinal mucosa till late-stage tissue infection of vital organs. So to this end, we aim to combine a liver-on-chip model with an intestine-on-chip model and try to see whether we can mimic the initial invasion, translocation through the bloodstream, and then invasion of vital organs. Of course, these projects, they also lead to tremendous collaborations where we can start looking at novel therapeutics, but also novel immunomodulatory therapies. Organ-on-chip technology in our lab has been presented, of course, at international meetings, and since then we have been contacted by several researchers from other institutes who show interest in this technology. For example, a guest researcher visited our lab for 6 months to learn more about this model, because they traditionally use animal in vivo models, but they also are now looking for alternative approaches for their research questions. Also, most recently, we obtained new research funding where we specifically will implement organ-on-chip technology in collaboration with researchers from Denmark to study novel therapeutics for invasive Candida albicans infection.